Welcome, everyone. As you can hear, we are recording. Welcome to the second ACID webinar on modeling on simulation. As I said in the last webinar, we want to use this webinar series to really showcase the capabilities and the know-how we have at ACID and partnering universities and companies here. So today it's very exciting to have two speakers. One from BOKU and ACID. We have Özge here, and also we have a speaker from a University of Vienna from Jürgen Zangelini's group. We have Leopold Zehedner here, and they will talk about metabolic modeling in biotechnology today. So a little information about the webinar and how it's structured. So Özge will start with a 20-minute presentation, then we'll have 10 minutes for questions and answers. Then we will hand over to Leopold, another 20-minute presentation and 10-minute Q&A. The meeting is recorded. For the questions, please use the Q&A tool at the bottom. You can either write your questions, they can also be written anonymously, or you can raise your hand. I think so that we can call you in to ask the question on the floor. Then after the first hour, we'll stop the recording, but we can continue the discussion if there is interest. And then also there will be a separate Zoom channel where we can have breakout rooms in case you want to have a one-on-one discussion with any of the speakers. So with that, I will hand over to Özge, who is here at BOKU University and also at ACID.
Thanks a lot, Nico, for the introduction, and thanks for the opportunity to present our work. Let me share my screen with you. Now, can you see it?
Yeah, looks great.
Perfect. All right. So as Nico said, for the next 20 minutes or so, I'm going to talk about how we use metabolic modeling and especially 13C metabolic flux analysis for exploring the yeast metabolism. And to give you kind of a heads up about the yeast that we are working with, it's Commangatia lapaffi, also known as Pichia pastoris. This might be the more popular name. It's an unconventional Crabtree-negative yeast, and it's an established host in biotechnology because it has characterized strains. The genomic sequences are available, and genome editing tools are also available, which makes it quite straightforward to engineer the cells according to our needs. Plus, it can reach to very high cell densities, especially the fat batch fermentations, which increase the titers of the product that we are interested. So this is another advantage too. And another unique feature of Commangatia lapaffi that it's a natural methylotrophic yeast, which means that in addition to other carbon sources like glucose or glycerol, it can also assimilate methanol via the xylulose monophosphate pathway that is localized in peroxisome. So in that respect, it can use methanol as the sole carbon and energy source as a single carbon source, but I'll come to that later on. And to kind of a short info about the method that we are using for the metabolic flux analysis. So you might be already familiar with it, but very shortly, it's a quantitative determination method to analyze intracellular fluxes in the metabolism and also the distribution of these fluxes in the metabolism as well. So what do we do if we have a carbon source like glucose, for instance? This is generally labeled with a carbon isotope, a 13C carbon isotope, because it's stable and it's safe to use. And then we introduce this substrate to the cells and then grow them until they reach the metabolic steady state, where we can have this unique profile of the isotopologues of different metabolites. And afterwards, what we need is a metabolic model that includes the carbon transitions of each reaction to show at which position the carbon goes to a specific position in the other metabolite. And then what we also need some extracellular constraints that we can measure, like glucose or substrate uptake rate or product formation rate, biomass formation rate, etc. And then by using this known data, what we do by iterative fitting, we try to reduce the error between the simulated and the measured values of this metabolite isomeric distribution, and then try to figure out the flux rate. So in a nutshell, this is how it works. And this we can do either by forward labeling, which means that we first grow the cells on a condition just to increase the biomass. And then afterwards, we switch them to a condition where we introduce a labeled substrate. This would be glucose, methanol, formic acid, CO2, and so on. And then after the cells reach the steady state, we then quench and extract the metabolites. And then generally, MS-based analytical instruments are used. Or what we can do, we can also use a reverse labeling method in certain cases, generally for practical purposes. What we can do also first, we can grow the cells on the labeled carbon, glycerol, for instance, in a condition that we are not interested to analyze to begin with. But the idea here is then to grow the cells and label all the biomass and then switch to a condition where we introduce our interest of substrate. This could be again methanol or glucose even, or carbon dioxide. And then the rest, the analytical part is then basically the rest is the same. And all of these MS-based analytical strategies are done in collaboration with the Institute of Analytical Chemistry at Bochum. All right, but then how do we use these tools for strain engineering or in general the explore yeast metabolism? So this was a work that has been done some time ago in our lab. And here the idea is to increase the production of a protein of interest. In that case, it is peroxide dismutase from human. And the idea was to determine the targets in the metabolism for overexpression or knockout for a more rational design of a strain. So the first metabolic flux analysis was conducted, and then this metabolic flux analysis showed the differences between the control strain and the producer strain. And for the sake of time, I'm going to just summarize, the key results. And this metabolic flux analysis revealed that there's an increased TCA flux and also a higher NADPH demand in the production strain, which shows that the cells need more energy. And this is not very surprising because protein production is a quite energy-intensive process. But with this metabolic flux analysis, we could show that, yes, this is also the case. We can predict it. But now the idea is, okay, then what do we do with that information? Then how can we determine what targets to delete or overexpress? And to do that, we had to introduce some more constraints to the model that we use. And in that case, it was a genome scale model of Comagata-Lapaffi. And to predict overexpression targets, this constraint was to maximize the product formation rate as an objective function, as you would call. And also the knockout targets, the idea was not to compromise the growth. So while keeping the growth similar to as the wild type, then deletion the unnecessary pathways that leads to the byproduct formation. So what happened afterwards, after running this analysis, some overexpression targets were determined. You see in the black arrows here, and these were generally in the oxidative pentose phosphate pathway and also in the TCA cycle, basically to increase the NADPH pools, obviously. And for the knockout targets, then in alignment with our goal, they led to an increase of the pyruvate pool here, so that most of the flux could be channeled to TCA cycle. Again, to increase energy demand. The only exception here was the PDA1 deletion. Obviously, this does not work because Comagata-Lapaffi cells needs the TCA cycle. So then the PhD student back then, what she did, she overexpressed or knocked out targets, And long story short, what she could show that most of the targets for the overexpression knockout actually led to the expected result and increase the protein titers at the end, which showed that how this metabolic modeling can be combined with a model-based strain engineering for improved recombinant protein production. Now I'm going to shift my focus a little bit from glucose to other carbon sources that I mentioned at the beginning, so-called the single carbon sources. So now the chemical technologies generally use the carbon sources that are fossil-based, can be also agricultural waste products, but unfortunately, these cannot be sustained for a longer time because the fossil-based resources, of course, with the climate crisis that we are facing these days, are not sustainable to use. Plus, also the other carbon sources which rely on the land use, they also compete with the agricultural production. So that's why the chemical technologies have to shift to some extent, at least, from fossil-based resources to renewable resources. And in that case, it's the single carbon sources like carbon dioxide, methanol, formates, or methane. And in that respect, I was a part of the Lighthouse project that has been done in ACIP. And our goal in that project was to produce some commodities or chemicals by the direct conversion of CO2 with an intention to develop a carbon sink so that we can contribute to the reducing of the greenhouse effect. So very shortly, the strain that we used was generated by a former PhD student from our lab, Thomas Gassler, where he converted the xylose monophos pathway. And by doing some genetic engineering, he integrated a synthetic CBB cycle or Calvin cycle to the cells. Now, the synthetic autotrophic Common-Gatella-Puffy cells can grow on CO2 using the sole carbon source and harvesting their energy from methanol. So this was our base strain. And the question that we asked afterwards in that project was that can we use this strain as a platform to make the chemicals directly using CO2. And we had two model products in that respect. One of them was itaconic acid and the other was lactic acid. They are both organic acids, which are produced in bulk amounts, and none of them can be produced natively by Common-Gatella-Puffy. That's why we had to integrate at least 3 genes from Aspergillus teres to Common-Gatella-Puffy. CADA is the key gene to produce itaconic acid from cisacotinates, and then also in addition to 2 transporters, MTTA and MFSA, to convert the cells to an itaconic acid producer strain. And for lactic acid, it sounded relatively more straightforward. It needed basically 1 gene, that's lactohydrate dehydrogenase from Lactobacillus plantarum, so that now the cells can produce lactic acid. So since it's not the scope of this talk, I want to show you that the results that can we make chemicals using CO2? And the answer is yes. Here on the left side, you see the itaconic acid production profile, where the cells can produce itaconic acid using CO2. It required a lot of metabolic engineering to balance the expression of different genes, but eventually we could produce. And also for lactic acid strain, that was also the case that we could produce lactic acid. Again, it required some more unexpected or unplanned metabolic engineering to be able to achieve that. But yeah, we can talk about it later if you're interested. But now the question here is that we ask, okay, we see these products that are being produced, but is it really the carbon dioxide that we are feeding to them to produce these products? Or in other words, where does the carbon come from? So to understand this, one way is to label the carbon and then follow it and see where it appears. But actually, this steady state labeling that I mentioned before is somehow limited when we are using single carbon sources, because you might remember this picture I showed you before. If we are using a multi-carbon source and we label just 1 carbon of it, or 2 or 3, however you design your experiment, at the end we have this unique metabolite isotope log distribution. But if you are using a single carbon source, basically what happens at the end when the cells reach to a steady state, everything is labeled because all carbon atoms are derived from the same source, and we cannot get enough information from the steady state phase. So to address this problem, what we used is in-station metabolic flux analysis. So this has been out for a while, but what we enabled this tool to be able to use in yeast biotechnology when they're growing on single carbon sources. So difference here to the steady state flux analysis is that in the metabolic flux analysis steady state, when isolate distribution which is reached to steady state, then we take the sample. This is how it happens. But as I said, this doesn't give us enough information when a single carbon source is used. But before this has reached this isolate distribution at a steady state, so-called isotopic steady state, there is a time period where a unique labeling pattern exists. So if we take samples, multiple samples at this time period, then basically we can have a unique flux solution by using the changes in this metabolites isotopologue profile. Of course, it comes with some challenges because it is more experimental work with multiple sampling points. You have to decide when to collect those samples, and you need rapid quenching methods, and generally computational times are longer. But this doesn't stop us, obviously. Actually, we use this method to answer the question that I asked before: where does the carbon come from in this itaconic or lactic acid strain? So what we did, we designed a reverse labeling experiment. As I mentioned before, we first cultivated the cells on labeled glycerol, and then after all the biomass is fully labeled, we inoculated them to main culture, where we feed them with unlabeled CO2 and also labeled methanol, because they still need methanol for the energy. And we took samples at different time points and then evaluated the results of the isotopologue fractions. And to do that, before the experimental analysis, we also calculated the predicted or theoretical isotopologue distribution and calculated the labeling degree. So I have to say here, since what I mean by labeling degree is that since we are feeding them with unlabeled CO2, we are washing the label out during the cultivation time, right? So when I say labeling degree, it's the reciprocal proportion of the CO2 incorporation. So anyway, by using a relatively small model that includes the CO2 capture and the glycolysis and the TCA cycle, we modeled this theoretical isotopologue distribution and calculated the labeling degree as 77 and 82% for itaconic and lactic acid respectively. And then very nicely, the experimental isotopologue distribution also fitted to the theoretical distribution, where it was 82% and 90% for itaconic and lactic acid, which was actually higher than we anticipated. There might be several reasons for that. Or obviously, one reason that we used a relatively small model, which did not include all the relevant pathways. Plus, there might be some other pathways that leads to the utilization of methanol or CO2 in that respect, which then changes the results. And actually, since we don't have a lot of time, this was one of the reasons for why this prediction doesn't fit like 100%. Because it was then turned out that the common pathway have some undiscovered methanol utilization pathways. So xylose monophosphate pathway is not the only metal assimilation pathway. There are multiple ones, and the PhD student from our lab, Bernd Mitic, did some strain engineering and created and designed experiments to show that actually common pathway has a reductive glycine pathway, which enables it to use methanol, formic acid, and CO2 simultaneously at the same time for growth and biomass formation. So this was another story, but it's actually a very nice example how these metabolic models can also lead to us to less unknown territories of the metabolism. Then finally, I want to talk about another project that we incorporated this stationary metabolic flux analysis to show you that this method is not only limited to single carbon sources, but also multiple carbon sources can be used for that. So again, in that respect, we had another PhD student, Evelyn, where she was integrating a carbon conserving pathway, a phosphocatalysis pathway for the acetyl-CoA production to Comagene Lepofi. The idea here is that to increase the carbon efficiency in the Comagene Lepofi for the production of citric acid. And she integrated this pathway to Comagene Lepofi, and it worked fine. She could show that this stress can produce more citric acid and so on. But now here the question, to what extent acetyl-CoA that is produced in this engineered strain comes from this the synthetic pathway that is integrated? Because also Comagene Lepofi, other pathways that lead to acetyl-CoA production can come from the lower glycolysis, can come from the pentose phosphate pathway as well. So to answer that question, she did the bioreactor cultivations with labeled glucose, and she sampled at different time points, and then from these samples, the acetyl-CoA that is produced is analyzed. And in that project, we used a bigger model that represents the metabolic metabolism more precisely, and then showed that how the fluxes are distributed. And actually, it was a very nice way to show that how the flux distributions are different in the control and the production strain. The first one, the control strain, the primary contribution comes from the glycolysis here in the blue line here, and the pentose phosphate pathway. Here, but in the producer strain, the primary contribution actually comes from the pentose phosphate pathway and also the GAF-6 pathway, that is the synthetic pathway that Evelyn integrated. And what we also saw that the contribution of the GAF-6 pathway was increasing over time to compensate the overproduction of citric acid that relies on the acetyl-CoA that is derived from this pathway. So with that, I'm already at the end of my talk. So just some take-home messages that the metabolic modeling and the 13C metabolic flux analysis helps us to achieve more rational and model-based strain engineering. And it can also helps us to uncover the unknown pathways. And saying that the continuous improvements in the metabolic models are crucial for enhancing predictive accuracy because there's still so much unknown, and integration of these pathways, especially the relevant pathways to your process, to metabolic models, increases the predictive accuracy, and also integration of the compartmentalization, especially in eukaryotic cells where parallel reactions can occur at different organelles, is another important parameter to consider. And finally, the instationary metabolic flux analysis enables us to explore single carbon metabolism in biotechnologically relevant hosts, not in yeast, obviously, especially when the use of steady-state metabolic flux analysis is limited. All right. With that, I want to thank to everyone who contributed to all of these works, years of work, and especially to Dieter Matonovic for giving us the opportunity to conduct this research. And then thank you for your attention. So if you have any questions, I'd be happy to take.
Thank you. It's a very interesting talk. Do we have questions from the participants? So actually, when you performed the instationary flux analysis, so how many more samples do you need to get the same information? Like how much more effort is it?
Of course, it depends to what you want to see and how fast the cells grow, what is the product formation rate, biomass formation rate, but at least like 3 to 4 to show that you have a profile. But also here, another parameter that is important, as I said, the growth rate and the product formation rate could actually affect how fast you should take samples and how often you should take samples as well. So generally what we do, we make kind of an first in silico experiment to model to make an educated guess where to take the sample, so that it's important that we know the characteristic of the cell, like the growth rate and the product formation rate, substrate utilization rate, etc., so that we can determine the exact time points that we should collect, that we can get enough information.
Okay, so you actually inform your measuring plan with the model.
Exactly.
Okay.
Exactly. Because for each metabolite, there is a different time period where you see they reach the steady state. Because for some metabolites, they are more closer to the pathway that you are working with. Could be in the upper glycolase, and they are labeled faster than the ones that are on the downstream. So that's why you have to know when to collect samples depending on the metabolites that you're interested.
Right. Okay. We do have a question from Barbara Petschacher. How do you do the quenching? What are the critical issues here?
Yeah, it's a good question. I have to say I don't have in mind actually the methods, but it's a method based on cold methanol quenching, and you have to be fast. This is the most critical thing, that you have to prepare all of your solutions and so on. But then the quenching depends how fast you are. So that's why it's important to work in parallel. And also another thing is that how much sample you take. Generally, what we are told that 10 milligrams of dry cell weight of biomass is enough, while we can go a bit lower than that to have reliable results from MS. But generally, that's the range, 8 to 10 milligram of biomass. And the other is that you have to be fast. I hope that's answered the questions. But if you're interested, then of course I can have a look and share our protocol with you. It's already out there. That's for yeast, for Comagetella puffy, of course.
Okay. Any more questions? So you worked mostly with smaller molecules, with commodities, right? You never. So, but Pichia or Comagetella puffy is being used for production of proteins as well, right? For biologics.
Definitely.
Do you know, is it then usually, is the carbon source then still glucose, or do people actually use methanol as a carbon source there?
Both like glucose or methanol. Yes, definitely this can be used, but it depends on the then of course when it comes to more like the industrial, like commercialization, then if the company does have to want to avoid from methanol, then of course glucose or other carbon sources come into track. But methanol can be definitely used for protein production as well.
I guess, yeah, methanol can be challenging for large scale
Exactly.
production, right? You
Exactly.
have it's both explosive and toxic. That's not the best
Yes,
combination.
of course, that we know how to handle it. It's, yes, you're right. It's explosive and flammable, but still in industry, it's being used. So
I mean, there are solutions to handle this, right?
Sure.
You can do large scale
Exactly.
ethanol precipitation. So
exactly.
why not that too? Yeah. Okay. I guess there are no more questions. So thank you, Özge.
Thank you too.
Then
I'm gonna stop sharing screen.
Yeah, then yeah, for the newcomers, I will just point out again that after the final Q&A, there's a opportunity to have a one-on-one discussion with the speakers. Yeah, for that, we will post a link in the chat for a separate Zoom meeting where we can have breakout rooms. Okay, but for that, with that, I hand over to Leopold Zehitner from University Vienna, who is a PhD student at the moment, but I think that will change this year very soon. Okay, yes, Leopold,
hopefully.
Please, you have the floor.
Thank you. Can you already see my screen?
Now we see it, yeah.
Okay, great. So, yeah, thank you for the, here it is. So thank you for the nice introduction. I'm going to present a more specific topic on modeling AAV production using HEC 293 specific genome scale metabolic models. So it's less broadly spread than the last talk, but more specific to a single bioprocess. This is part, or the main part of my PhD project, and the paper was published, I think, last week, so it's hot from the bench results. And if you want to check out, the preprint is already available on bioRxiv. So what's going on during the next 15 to 20 minutes? There will be a short overview of gene therapy. What is it? Why is it important? And what are the limitations? Secondly, I'll again introduce genome scale metabolic models, although Özge already did a great job in explaining that. And the final part will be the main results from my PhD topic and paper. Okay, great. So let's start with that. So what's gene therapy at all? In gene therapy, you normally have some vectors, or you normally distinguish between two forms: in vivo gene therapy and ex vivo gene therapy. In vivo gene therapy means that you got some vectors, viral vectors or lipid nanoparticles or so. You put your gene into the vector and then you apply it to the patient so that the gene is delivered to the target organ and can the gene can then be expressed again. In ex vivo gene therapy, it is a bit different because you extract cells first from the patient and then you freeze them with a viral vector, so you transform them. So just as you know, for example, for CAR T cells, you expand them in culture and then you reintroduce the cells into the patient. Both fields are currently summarized in the cell and gene therapy sector, and both are super hot topics currently because, as you can see here, the approved viral gene therapies by FDA are still, oh, it's okay, 10 already, with 7 having AAV as viral vector to deliver a gene, but it's still in very, very early phases of this field. However, the prediction for industry are quite good, as you can see here with AAV manufacturing predicted growth to approximately over 50 billions of US dollars, and also the academic interest is quite high, as you can see in this exponential rise for the published publications. Yeah. Background for, because my topic mainly was the optimization of an AAV production process. So I would like to introduce you to what is happening and what's going on, what are the challenges for producing an adeno-associated virus. In general, you have the most common process is to have 3 plasmids. You have your nanoparticles, they're taken up by the production cells, come into the nucleus, and there the genes on the plasmids are used to produce the AAV. Here starts the first challenge because you always have some particles that are filled with the target gene of interest and some are empty. And what you of course want is not applying empty particles to the patient because they're useless. They cannot deliver any gene. After some PTMs and modifications, you normally secrete the cells secrete the AAV, and they can be purified from the broth. As in every bioprocess, you need to feed amino acids for the production of the AAV and glucose and so on for the energy supply. What is very important for the gene therapy market currently, especially for the viral genes, is that they have super huge potential because there are drugs where you can just apply 1 dose to a patient and a genetic disease is cured, such as Zolgensma, for example, from Novartis. But this makes the drugs, of course, extremely expensive. So it might be that a shot costs between $2 and $3 million US dollars. and often the government does not take over the full cost. In terms of a bioprocess, you need approximately 1,000 liters of a batch process to treat one patient, which is also tremendously high. Of course, depending on the disease. For example, there are some eye diseases where you need less titers, but in general, that's quite high. So we got a lot of challenges here. We got the titer challenge, the scale-up challenge, also some downstream purification stuff. But for that, yeah, I'm not the best person to address these questions, and maybe, yeah, in the last webinar there might have been more answers to that. Yes, so what did we use? We used genome scale metabolic models. What are these genome scale metabolic models? These are constraint-based models, so they are representing the metabolism of a certain organism by using biochemical reactions. And in constraint-based modeling of these genome scale metabolic models, you have always a steady state assumption. So the steady state assumption means that in contrast to kinetic models, you have not a time resolved manner, but you really have snapshots of your organism. You add some exchange rates that you calculated, or you add your current biomass composition, and then you or add other flux bounds to reduce your solution space. And then you optimize for a specific target, for example, the biomass production or the product formation. And this gives you then an idea of which pathways or which fluxes or reactions are more active than others and differ between conditions or between cell lines or whatever. So this field is currently also quite evolving to compare, for example, also disease states versus non-disease states. Yeah, but the perfect fit for this topic is, of course, the bioprocess, because you have a controlled environment and you can sample your environment, and therefore you get pretty nice data. And yeah, my colleague Diana, so now a supervisor, but during her PhD project, she discovered that the most important types of constraints are the exchange rates, so what's taken up by the cells and what is secreted by the cells. And what is not that important is the biomass composition. However, also transcriptome data, enzyme, proteome data, or enzyme data can be incorporated into these models to get a more refined understanding of what's going on in these cells. Okay, so since this is clear, I would like to give you a short overview. This is from our review published 2 years ago. What is the current or our most widely used approaches for strain design? Currently in industry, it's just high throughput passaging. So you repeat passaging and adapting cell lines to your specific medium or condition or bioprocess. And after some time, they grow faster, they produce better, and so on. What is also very common is just to do a labor and time intensive DOE, so checking different conditions and then selecting, okay, which is the best combination of certain metabolites, temperature, pH, and so on for the yield. What is bit more sophisticated is the single omics or hypothesis driven approach when you, I don't know, invest tons of hours of reading literature or doing transcriptomic experiments, for example, as my colleagues at working on this project before did, when they just used transcriptomic data to analyze this AAV production process. Yeah, but it does not always give you the full picture of what is happening, especially regarding the metabolism. And here's just an overview of which host cells are currently used for the production of the most important viral vectors. So we have, of course, the most important is the HEC293 cell, because it gives the viruses the human glycosylation pattern, and therefore it's not so. There are also strains from other origins, but the workhorse in this very field is the HEC293 cell. Yes. So what was our approach? Our approach on the up here is we did some fermentation. We took samples for the multi-omics measurements. Based on these data, we reconstructed context and time-specific genome-scale metabolic models. After simulation, we identified a target, and this target was then inhibited to validate the simulation results, also experimentally. So in more detail, that what I would like you to understand for the following slides and the results, we had two strains. We had a high-producing strain, which was cultivated over 10 years, I think, and passaged through and through and through. So this was super optimized for AAV production in a specific medium, and one low-producing strain that was just adapted from a adherent HEC293 cell. So this was quite bad, but the reason why we did that and not just focus on the high-producing cell is just the licensing cost. Because if some research institutes or so put tens of or decades into optimizing a strain, they would not like you to have them for free. Secondly, both of these strains were used in a transfected state and a mock state, so mock without any plasmids transfected, to compare the transfection states or the production states. And then we took samples before transfection and at 4 time points after transfection. Normally, an AAV production process following triple transfection runs, for 3 days approximately. Some of them are mentioned for 2 days, some for 4 days, but the average in literature is normally 3 days. And we took several samples to measure the lipidome, metabolome, transcriptome was already measured by colleagues before. And we got context-specific and time point-specific metabolic models that we use for simulation. Okay, and now we start with our results for the biomass composition, because as I already mentioned, Diana worked on before this project, worked on a similar project in CHO cells, and she discovered that the biomass composition did not differ that much between strains. However, we did the same, and we discovered something completely different. So in the first plot you see here, in the first two, the high-producing strains, then here are the two low-producing strains conditions. And here for the reference is one CHO strain. And as you can see, the HEC strains differ in size and weight. So it's not with as with the CHO cells, the biomass composition did not differ. Secondly, the low-producing strain had a very much higher overall weight, but only when broken down to the single compartments, so or macromolecules, DNA, RNA, glucose, and so on, only the difference in the glucose or glycogen levels were significantly different between those two strains. However, when we focus more on the real lipid profile of these two strains, so here is now again the high-producing strain and here is the low-producing strain, I would like to point out one very important difference, because in general they are pretty similar again. But here you can see in blue an increase of the triglycerides in the high-producing strain. So the bars here are becoming bigger and bigger and bigger. So the high-producing strain is produces from the too much or accumulating glucose. It converts into triglycerides, whereas this effect could not be observed in the low-producing strain. And this matches perfectly to our results from the biomass composition on the macromolecular level, because the low-producing strain tended to store glucose as glycogen and not as lipids. Also, what you can see here is a PCA on the lipidomic data. And what is important to take from this picture is that the separation is not what we expected first to be the production state. So we expected, okay, maybe there should be a higher similarity between the AAV production and not AAV production. But in contrast to that, the signal or the difference between the two strains was more significant. So while you can see here the two high-producing strains, in orange it's transfected, in light blue it's mock, and they cluster more together than between the states. So for example, the transfected two transfection states have nothing to do with each other. Secondly, from a metabolic modeling perspective, it's important to just give you a short introduction, or sorry, I have to shift the window so that I can see the graphs again. So here are the cell growths of these high-producing and low-producing strains from my colleagues at Takeda before that, published by Martina and Caroline. What is important for the modeling of the production process is that we are not in an exponential phase anymore. And this is, of course, should be normal for a bioprocess now. But the main issue is then the modeling, because normally you use to predict you use the exponential phase assumption. So you got one growth rate over the whole time. And what we can see here is that the growth does not behave exponentially, but in contrast, it's sigmoidal kind of. So when comparing the simulated growth rates from our models to the experimental growth rates, it was a bit tricky because we needed to fit another curve than an exponential curve, and this led to way higher standard deviations and variation within certain time points. However, we could show that the predictions of our models from the simulated, or the simulated growth rates from our models are pretty okay, and are also in accordance with literature, for example, because this is another HEC process where the exchange rates have been measured from literature in an exponential phase. So our metabolic model, our cells and the simulated growth rates behave pretty similar and are in a similar range regarding the growth rate. So this was just used to really validate, okay, we did not, or we reconstructed valid models and did not do something implausible. Here is a more interesting plot, maybe because it's not just focused on growth, but on production. So in blue, it's the low producing productivity, and in orange, it's the high producing strain and the production as a function of the maximum growth. And what is important to mention here is that both strains have a maximum production rate of 10 to the power of -9 or so, something in this area. And if we would, and that's what we simulated here, if we would completely stall growth and put all our resources that we provide in the bioreactor put into production, a titer between 10 to the power of -7 or even higher would be possible. So we are, even in the high producing strain, we are not using the full capacity of the cells. We are just using below 1% of the resources for production, and over 99% of the substrate, so amino acids, glucose, etc., is just used for growth, which is from a various aspects. So not just a financial aspect, but also from a sustainable aspect. Very, very critical. So what we did is we simulated our metabolic models over time. We used a random forest classifier to compare the different flux states and so on. But I would really suggest to read the paper if you're interested in that. I would like to focus now more on the results, because we identified, based on the fluxes, a transcription factor called HIF1 alpha. This is one of these master transcription factors, mainly studied in cancer cells, because there you have a state of hypoxia, which means that the cells are not supplied with enough oxygen anymore, and they need to shift from aerobic metabolism to anaerobic metabolism. However, in a bioreactor, you would not have assumed this behavior, because you supply your bioreactor with enough oxygen and you put in their oxygen as much as you can. So normally you do not go below thresholds like 20% of dissolved oxygen. So interestingly, we found this transcription factor to be active from our flux sampling approach. And here's just a short overview of what this transcription factor can change in the metabolic profile. It can lead to higher glycolysis, of course, because the cells shift to anaerobic metabolism, and it increases the PPP. It shifts the NADH to NAD ratio, so the energy metabolism, to a reductive state, to a NADH accumulates. It also increases the production of reactive oxygen species, certain oncometabolites, and it increases glutaminolysis. Glutamine was, of course, added, as in every common mammalian medium. Glutamine was added to the medium. And it also, of course, increases the unfolded protein response and the ER stress, secretion of extracellular vesicles, and activates the certain immune genes. So this fully reprograms the cell metabolism and behavior. And what we did is to add a inhibitor to these cells. And what we could observe is that we completely stalled growth in these cells. So we could show, okay, in this very state, the low producing cells are fully reliant on this transcription factor, although enough oxygen is supplied. However, we did also do some from the spent media analysis, we found some limiting components, being amino acids, so aspartate and glutamate. We added them, and there was no change in overall growth. So this did not change anything. What is important for the production is that while the AAV capsid production increased by 250%, the viral genome production decreased. So when comparing the reference samples here, the viral genomes were decreased in these inhibited cells, and this might be due to the reason because there was either not enough glucose uptake or we really inhibited the PPP. But yeah, there is much further research is necessary to really find the causes. One, of course, thing would be to decouple the PPP from the HIF1 alpha to really have an independent nucleotide production, and then the viral genomes might also increase again. What is important for you now to take away compared to the project of before using just transcriptome-based studies? You got 40,000 genes. So I worked with the same transcriptomic data to refine my models. You get differentially expressed genes, and they are grouped in gene ontology terms. So whenever you already worked on transcriptomic data, you get these big lists and so on, and you can use fancy models and statistical models. However, the main issue is that with over 40,000 genes, the significant ones might be misleading, and you always, or it could be the case that you don't find the needle within this within these 40,000 genes. So, for example, they found the one part of the interferon pathway, the STAT1 gene. They inhibited it, and they got an increase of 200%, but only in shaker, so not in the bioreactor, which was also quite interesting, and they did not expect that. Here is the transcription of the STAT1, because normally, or what we thought would have happened is that, okay, after transfection and virus production, the cells might increase the immune response. But as you can see here in orange, that's the low producing strain, and here in blue, it's the high producing strain. And even before transfection, the expression of STAT1 is already higher. So there is already the low producing cells are already in a bad state before transfection and not after transfection. And what we could show, so within these 40,000 genes, they could not identify HIF1 alpha as important transcription factor just due to multiple test correction. And with our approach and scaling down to 8,000 reactions and 2,000 metabolites, we could identify the real cause of the state of the transcription factor. Yes, this leads me to somehow still, or I have to mention some limitations, because with identifying this transcription factor, we need to question the approach if we want growth coupled production or non-growth coupled production, as I already mentioned in this one slide, where resources are maybe not used in the best way. If you imagine, okay, just under 1% of the resources go into production. However, you need to consider that normally for complex biologics such as viruses or recombinant proteins, the growth coupled production is the go-to approach. However, we need to reconsider that maybe if you really find a transcription factor where you can control your cellular growth. Yeah, finally, I would like to thank my the group at the University of Vienna, especially Diana, Jürgen, and Juan. Also the group from Takeda in Orth. And I would like to do some obvious ads for our upcoming conference, the MPA in Vienna 2025, summer school before the conference, and of course we have 2 open PhD positions in modeling of bioprocesses and bioengineering. So if you're interested in that, please visit our group's webpage, and thank you for your attention.
Leopold, a very interesting talk. We already have a question from Larissa Hofer. She asks, what kind of model are you using, and is it specific for HEX cells?
It was so there are global reconstructions for all the metabolite or all the reactions in the human cells, and you can use transcriptomic data and certain algorithms to create context or cell line specific metabolic models. So for example, it is known that I think 25% of the genes are only expressed in the nervous system. And since we worked on kidney cells, these 25% are not necessary. And you can use transcriptomic data to really get only those reactions that are based on the expression profiles more likely to be active. And what has been shown in literature is that when you use context specific models, the prediction accuracies of your models really increase. So it's worth going for that.
Okay, I hope that answered the question, Larissa. Are there more questions from participants? Actually, I have a question. So you mentioned that it's less than 1% of the resources that the cell is spending on the production of AAV
Yes.
in this case, right? Right, that sounds very low to me, but then again, I don't really have a comparison. Do you have an idea of what those numbers look like? For example, if you produce a monoclonal antibody in CHO cells, I mean, here we pretty high titers can be achieved, right? So
I mean, yes, that's true. I'm not an expert in recombinant protein production, but I would, since these processes are normally also growth coupled, and there is also always a limit around, I don't know, between 6 and 8 grams per liter. I would expect that there is more possible. So we have also some space for improvement there.
I mean, I assume that with 1% that the ceiling really has not been reached yet, right? So there must be a lot of room for improvement. So you looked at AAV specifically, right? But
yes,
there are other gene therapy vectors out there. Some of them also envelop viruses. Would this model change significantly if you looked at the production of the herpes simplex virus-based gene therapy vectors that are approved right now, or could you apply the same model?
I would not expect that it would change a lot, since you just need to consider additionally or parts of the lipid profile that are also used from the cells. But the approach in general can be applied easily to, for example, retroviruses or enveloped viruses.
Great, yeah. And so this HIF-1 alpha inhibition, do you think there's a way to fine-tune it to increase the production, not just of capsid, but of, well, fully assembled AAV?
Yeah, sure.
Because I mean, there was full inhibition you tried, right?
Yes, exactly. So what would be or would be interesting to test if we, however, somehow either not fully inhibit it, but just knock it down, so that the PPP stays somehow active, or to supply the cells with nucleosides so that the production from the PPP is not necessary anymore. I mean, what we did not expect is that we could inhibit mammalian cells fully in a bioreactor by just inhibiting one transcription factor. And that was, changed a bit the next steps that we were thinking about.
Okay, very interesting. Yeah, thank you for your presentation. I will just announce the next webinar we have planned right now. So yeah, I hope you can join us in a month on April 28th. We have two speakers, Barbara Petschacher from ACIP and TU Graz, and Peter Satzer from BOKU. They will talk about sustainability in bioprocessing. Yeah, so same time in a month. Yeah, and please join us if you want to have a chat with Özge or Leopold. Please join us. Click the link in the chat, and then you can have one-on-one discussions or discuss if you have any questions. So yeah, thank you everyone, and maybe see you in the breakout.